Good evening, everyone. Welcome to tonight's Lung Cancer Living Room. All of you in the room, all of those joining us from online, welcome, welcome, welcome. It's great to see some familiar faces in the room and great to see some new ones as well. For those of you who do not know me, I am Danielle Hicks. I am the Associate Executive Director overseeing programs and services here at the Foundation. I am also one of Bonnie's daughters.
The oldest.
The oldest. The youngest is in the room somewhere, I think. I don't know. Anyway, we're thrilled to have you guys here tonight to talk about targeted therapy resistance. It's a huge up-and-coming field right now. There's lots of really exciting things happening. So we are thrilled to have Dr. Collin Blakely here from University of California, San Francisco, and Terry Kennedy, who you will meet for those of you who have never met her. She is a lung cancer survivor, and she's going to share her story with us. So thank you.
Now I'd like to introduce Dr. Collin Blakely from UCSF, and he's going to talk to us about progression and resistance and why all of those things are happening.
Great.
And what we can do about it.
Thank you so much, Bonnie, and thank you to you and your organization for inviting me. You know, you are definitely a legend in the Bay Area and now nationally, and I'm just thrilled to be here, so thank you so much. And, you know, I'm inspired by all the stories. Many of you have been fighting lung cancer for years and some even decades, so it's about you. That's what makes me do what I do, to try to help you and to improve the therapy. I think if we just stick with the status quo, it's not enough. And I've kind of dedicated my whole career, much of my life, towards improving lung cancer treatments and coming up with, hopefully, some of the next therapies, or at least being involved in that. So today I'm going to talk about overcoming resistance to targeted therapy, in particular in lung cancer. So over the past decade or so, we've begun to understand lung cancer, in particular non-small cell lung cancer, and I would say more specifically adenocarcinoma at a much more defined molecular level. We've been able to determine that there are key mutations within the genetics of the cancer that seem to be critical for driving the cancer's growth. They're addicted to these genes and these mutations. And as such, if we can find drugs that can target them, in many cases we can cause these tumors to shrink, in many cases shrink dramatically and for long periods of time. I would say that's been most successful with EGFR, as some of you have talked about, as well as ALK, which makes up a smaller piece of this pie, but still very important. And there's been now at least 3, and actually 4 FDA-approved drugs to target EGFR, and another 4 to target ALK. And these have been huge advances. However, these treatments almost always stop working at some point during the disease course. How do we overcome that? So first I'll focus on EGFR, and that's really my, I would say, area of expertise and what I spend most of my time studying, although I do have interest in ALK and other targeted therapies as well. But EGFR stands for epidermal growth factor receptor. So it's a protein made by the gene that goes to the surface of the cell, and it responds to signals from outside of the cell and tells that cell to grow and divide. And it's a very important gene, a very important protein. It's made in almost all of our cells in our body. However, when you get a mutation in that protein, the cells no longer need that cue from the outside. They no longer need a signal from another cell saying, okay, it's time to grow. And they just do it automatically. It's basically like a light switch, you just can't turn it off. Fortunately, we have developed drugs that in some cases can turn it off. And those are effective most against these L858R and exon 19 deletions, which make up the majority of EGFR mutant lung cancer. However, as many of you know, and many of you have actually experienced this, at some point, these drugs stop working, and we get what's so-called acquired resistance to therapy. And that's when you move on to new drugs such as Tagrisso or chemotherapy or immune therapy. What we're finding, however, is that that's a harder and harder disease to treat because there are not just a few mutations, say EGFR maybe plus a handful of others that are driving the tumor's growth, but maybe there are dozens of mutations in the cancer at that point. Maybe there are what we call subclones present that have additional mutations that the initial tumor didn't have. So our focus at UCSF, and I'm partnering with Trevor Bavona, Dr. David Jablons, and others, is to understand what's happening at this initial response to treatment. Why is it that the tumor usually responds but doesn't go away completely? Definitely you can have complete responses to therapy, but I would say they're pretty rare. So why is it that the tumor is persisting? What can we find out about it at that point and maybe target before it even gets to the point of acquired resistance? And that's really the major focus of my work at this point. One of the ways to understand this better is to get access to the tumor DNA at pre-treatment, at this kind of residual disease state, and see how it evolves over time to become a fully progressing acquired resistance tumor. And we do this in part by doing biopsies of the tumor itself. But as you guys know, sometimes that's very challenging and can be painful, and sometimes we don't get adequate tissue to analyze. So what I've done is I partnered with Guardant Health, who many of you are familiar with, to do liquid biopsies on patients, and was able with them to get access to their database of about 1,200 EGFR mutated lung cancers, and to do a thorough analysis of what's happening at this, even before treatment, that might be influencing how well somebody responds to an EGFR inhibitor, what's happening during treatment, and what's happening at resistance. And what we found is that this cancer is much more complex than we had originally realized. So it was initially thought that, well, if we just find the one mutation that makes it grow, that's all we really need to know. But in reality, there are dozens of alterations frequently in these tumors. And we really need to know what's happening in every one of those pathways and what role it's playing in the cancer's growth and progression and resistance to therapy. So to do that, I looked to see how patients did, and I correlated it with what mutations we found in their plasma, in their what we call their cell-free DNA or circulating tumor DNA. And we found a number of mutations actually did correlate with worse response, in this case to osimertinib or Tagrisso, in different pathways. And I'll zero in on something called the cell cycle. And the cell cycle is what tells the cell to make a copy of itself. It's very tightly controlled by multiple proteins and pathways within the cell. And when that gets altered, the cell can grow out of control. We thought that maybe EGFR signaling was enough to do that, but it turns out if the tumor also has an alteration in one of these cell cycle genes, it's even more aggressive. And in this case, resistant to osimertinib in many cases in the patients that we've looked at in this study. So how do we potentially overcome that? Well, we're working right now to develop a potential new therapy. We're doing this in what we call preclinical models at the moment. That means in the laboratory, testing cancer cells in Petri dishes, and also in mouse models of cancer to see if this therapy will work. And we're working with drug companies to take this to patients to develop a new clinical trial and hopefully change what is the standard of care for treatment of this disease. And that treatment would be a combination of osimertinib with what's called a CDK4/6 inhibitor. So CDK4/6 is one of these key factors in the progression of the cell cycle. And we found that that gets altered in a lot of these cancers. Fortunately, in the breast cancer world, they've already developed a drug against that. So we're hoping to kind of repurpose those. Actually, 3 of them are now FDA approved. We're hoping to repurpose that drug for lung cancer. And it's started to be studied in lung cancer and it hasn't really had a lot of traction yet. But that's because I don't think they're studying it in the right population. They're just studying it in every patient. I think it's only in the patients that have specific alterations where you're going to see a benefit. And I think you're going to need to use it in combination with other therapies, not just by itself. So what I'm proposing to do is to use it in combination with osimertinib with the idea that we can shrink the tumor and maybe even get some of these what we call complete responses where the cancer goes away completely, with the idea that that's going to delay the time before the cancer begins to grow again. We're trying to basically kill more cancer cells from the get-go. Another way that we're focusing on this problem of disease persistence is to actually get access to tissue at that persistent state. As I mentioned, biopsies are challenging at that point, and it is putting a patient through a procedure that they don't need clinically, but really purely for research. And that is hard to come by, and many people, many patients have volunteered to have that done, and we're very grateful to them. But another way to do this is to do it in patients that are already going to have that tumor resected anyway through surgery. So I've partnered with AstraZeneca and David Jablons at UCSF. He's the chief of thoracic surgery there, for those that don't know him. And what we're doing is a clinical trial to identify patients that have early stage EGFR mutant lung cancer. This could be up to stage 3A, which means they have lymph nodes on one side of their chest. And we're going to treat them with Tagrisso or osimertinib for 1 to 2 months prior to surgery. And the expectation is that that's going to cause the tumor to shrink, but not completely. And so what we'll have left is this residual disease that happens in many patients that we don't understand. But Dr. Jablons then will go in and he'll remove the tumor, and then we'll have it to study. And we'll be able to do any of a number of things. We'll be able to look at the transcriptome, what's called the RNA of the tumor. We'll be able to look at what genomic changes have been selected for by the drug treatment. We'll be able to look at what immune cells potentially come into the tumor and either are trying to eliminate the tumor that's left over or are actually helping the tumor to survive. The tumor is very crafty in a way. It can recruit immune cells in to give it growth factors to allow it to survive inhibition of EGFR. So this will allow us to understand all of those things, and then once we understand it, then we can target it. Then we can come up with new combination therapies. And that's our hope that this study will.
What's the benefit to the patient? Because this is stage 3?
Up to stage 3. It could be stage 1 through 3.
Mm-hmm. But it's not really usual to do surgery on 3.
For us at UCSF, I would say it is.
Well, I know that because I was one of them.
Yeah.
But everywhere else, it's not really necessarily standard of care. So the benefit to the 2 months of therapy is, what is the benefit to the patient other than
so
research?
I think there's several possible benefits. One is that we could downstage the tumor because we think that 80 to 90% of patients with this EGFR mutation will respond to this treatment. The tumor will shrink.
Okay.
So we could downstage it from something that was borderline surgically resectable to something that's much easier for Dr. Jablons and his team to resect. So just want to, in the last couple minutes, switch gears to ALK. This is something I'm also very interested in. So ALK is it's not a receptor, but it's a gene fusion. So what happens in ALK is that you basically have the DNA in one part of the cell breaks off and connects with another part of the cell, and it's just not, these two genes are not supposed to be together. And what happens when that occurs is that you get massive expression or massive production of this protein called ALK. And that can signal through similar pathways that EGFR can to drive the growth of the cancer cell and turn it into a cancer. This was discovered now going on about 10 years now, and it's really been amazing the pace of drug discovery here. We had crizotinib approved in 2011, and quickly thereafter we had another drug, ceritinib, and then alectinib, and now brigatinib, and then we expect lorlatinib to be approved soon as well. So these are all potential drugs that can target ALK and cause tumors to shrink. However, like EGFR, the problem is that resistance still occurs, and these tumors don't go away completely. In many cases, we can identify the mechanism of resistance through what's called a second mutation in ALK that causes the cancer cells to become resistant to that targeted therapy. But as you see in the white there, about half of the patients, you can't find anything that's causing resistance. So why is that? How do we treat that? Well, one possibility is to look at how ALK signals. What are the downstream pathways from ALK? Maybe it's one of these pathways that gets altered that's driving that resistance. And ALK is known to signal through multiple pathways, what we call the PI3 kinase pathway on the upper, the STAT3 pathway, the RAS pathway, and the PLC gamma pathway, among probably a dozen others as well. Which one of these is actually important to this tumor? We didn't know. Until a graduate student in Trevor Bavona's lab did a very elegant study in which he used genetic approaches as well as drug approaches to systematically go through every one of these pathways and figure out which one ALK was truly dependent on. And what he came up with was that MEK pathway, that MAP kinase signaling pathway. And fortunately, there is a MEK inhibitor that's available. There's two, actually. One of them is trametinib, the other cobimetinib. And in a mouse study, he found that if he just used the drug ceritinib, which is one of these second-generation ALK inhibitors, by itself, he got nice tumor shrinkage for a while, but then after 60 or so days, it started to grow again. Very similar to what happens in patients, only on a kind of compressed time scale. But if he started treatment with ceritinib and trametinib from the beginning, all of the tumors were eliminated and never came back. So we've seen, these dramatic responses in mice before, so I don't want to get too excited about this, but it was really intriguing data. And to the point that I worked with Novartis over the last 2 years to develop a clinical trial to combine ceritinib with trametinib, and that study is now open at UCSF, and we're accruing patients to that. So if any of you are interested in that study, I'd be happy to talk to you more about that. So I think with that, I'd probably use my time, and I'll just thank you. And none of this work could be possible without you guys. So you volunteering to participate in the research is just tremendous. And thank you for that. And just want to thank the rest of my team. And Bonnie, early on, the foundation funded me back in 2012 when I was a fellow. So kind of started me on my way. So I'm very thankful to you for that.
You're welcome. Terry, your story, you thought you were going to get away without it, but you're not.
Okay. Like everybody else, I have a lung cancer diagnosis. Not everybody, but a lot of folks here. I was diagnosed in August of 2015, so coming up 3 years. I've had a lot of experience with resisting targeted therapies, so that's kind of unique. I've done a lot of lines of treatment, and that's why this is very interesting, and I can share my experience with trying to come up with ideas to deal with the resistance. So I started out after diagnosis and having I had a very large tumor on my upper right lung that I think was like the size of my fist, and progression or metastases to my bones and some lymph node. And so, because it was considered to be growing at a fast rate, before I even got the genomic testing results, I went on a chemo and did that, just a pemextred and carbo for 2 rounds. And in the meantime, I got the results that I was EGFR. So we went ahead and waited to see if I responded to the chemo. I really didn't respond. It didn't grow significantly, but there wasn't any shrinkage as a result of the 2 rounds. So we switched to the targeted therapy, which I, at the time, did afatinib shortly after diagnosis. And I was a super responder to afatinib. The tumor shrunk to the size of a dime, is from what I understand. And it went away in my bones and no spread. But I responded tremendously, but not for very long, like 8 or 9 months. And then new nodules started to appear.
Yeah.
And I never had that stable period. It was just like shrink and then, yeah. So after having a tissue biopsy after showing some growth after afatinib, T790 showed up, T790M. So I went on Tagrisso, and this was close to a year after being diagnosed. Moved to Tagrisso. One scan after being on Tagrisso, I think was okay. And then the next scan, and I do scans like every 6 to 8 weeks. I'm a pretty frequent scanner. And the second one shows some growth again. So I really did not respond that well to Tagrisso, which I think of another question. You talked about why some people don't respond to Tagrisso, and I don't know if that has to do with as a second line or a beginning line or what, but anyway. So I never know why I didn't respond to Tagrisso. But so after that, I had another biopsy which showed C797S, I believe. And the T790M went away. But again, it was maybe a different part of a tumor and that whole thing. So I became a clinical trial junkie after that, trying different things. And, I didn't think I wanted to do chemo. I was trying to push it off. I wanted the targeted therapy to work or some combination. So then I'm treated at UC Davis with Dr. Reese, and he was principal investigator of a trial that's been pretty successful with nestatumumab plus Tagrisso. And so I tried that, about a year and a half ago almost, and I had a very toxic response to the nestatumumab, a very unusual, and actually I don't have very many side effects, and I've taken a lot of different drugs, but for some reason, I don't know if I was allergic, but I had to drop out because of toxicity, so didn't really get an idea of how that would have worked for me, but
I just want to point out how unique and awesome it is what Terry has been able to pull together for the EGFR resistors, to be able to get that many similar patients together and agree to go out and be involved in research is a huge feat.
Yeah.
And our clinical trials, wouldn't be weighing as low as they were if more people were doing what you guys are doing. And there's groups out there for ALK as well, and for ROS1 and whoever else might be watching, interested in some of these groups that have pulled together. And we're working with all of them in some capacity, and it's just truly, truly remarkable. So thank you for that.
And we actually had a call with Collin and Trevor about the organoids, and we're not stuck on doing just one thing.
yeah, we're kind of feeling around right now to figure out what
to
get
look at other
behind,
options, and so but it's a clinical trial accrual is one of the biggest issues out there. So for these patients to be pulling together and creating these groups, you won't have to do so much fishing. They're going to be coming to you, and saying, I need this many. And, even into the submarkers, it's not just about EGFR, because you might say, I need 10 exon 20s, or I need, various different things. So.
And we feel like we're still trying to grow our community, and the more people we have, the more data available, and to coincide with a lot of what you're working on. And we know we're not all starting from scratch, but we could provide a lot of information about our treatment history and our mutations that would maybe drive some research. Sure. Oh, look at all this, the number of percentage of this showing up, and these people also took this, and some informative information that we're hoping to help.
No, that's great.
And will be. I have a question for you, Collin, because I'm just a huge believer in patients being a big part of the solution. And I constantly talk to everybody I have an ear to, that clinical research, we always call it from the bench to the patient. What do you think about transformational research, and call it from the patient to the bench and back to the patient?
Yeah, I think that's what we refer to it as. I agree with you that bench to bedside. I actually think for me it's the reverse.
Right.
I learn the most from the patients. I think I learned that early on in my training, that being in the laboratory, you can only learn so much from treating cells in the petri dish.
Right.
And where I was learning the most was from the patients I was seeing in clinic, and that's why I actually kind of switched from being a lab-based scientist to being more of a clinical translational scientist. Because I learned the most from the patients, and that's why, we did that garden study. We tried to learn, and you're exactly right. We need that information about how you did to the therapy that you were on. What other treatments you've had. It's a big puzzle to put it all together.
Right.
So absolutely. Completely agree with you.
And we have a patient registry where the patients can put their own information in for patient-reported outcomes. And as the registry gets bigger and bigger and bigger, eventually we'll be able to connect with the EMRs and validate the data. But data is the key. It's the key.
Absolutely.